one day multidisciplinary international e-conference on sustainable development goals, key issue, opportunity and challenges in environments, agriculture, pollution, resources, economy and social sciences. My title is Comparative Study of Antimicrobial Activity of Aloe Vera and Catharanthus roseus by Nidhi Rathod, Chemistry Research Scholar, Dr. APJ Abdul Kalam University, Indore. Abstract: the comparative study of the antimicrobial activity of aloe vera leaves and Catharanthus roseus flower extract against selected clinical isolated have been investigated using standard microbiologically method. Introductions: plants have been an important source of medicines for thousands of years. Aloe vera is an important medicinal plant of Liliaceae family. Species of aloe vera plants used in traditional medicines and most plants have show antibacterial activity. Aloe vera is a cactus-like plant with green, dessert-shaped leaves, data or fresh trapping, spiny, marginated and filled with a clear viscous gel. These images are aloe vera leaves. The name was derived from the Arabic aloe meaning bitter because of bitter liquid found in the leaves. It is known as lily of the desert, the plant of immortality and the medicinal plant with quality to serve as alternate medicine. Aloe vera possesses a number of therapeutic, anti-inflammatory, antibacterial, antifungal, and cell growth stimulatory activity. Examine the antimicrobial activity of aloe vera on human pathogens such as Staphylococcus aureus. The main objective for this study to determine the antimicrobial activity of aloe vera leaves. Catharanthus roseus is an important medicinal plant of Apocynaceae family. Its commonly name known as Nayantara or Sadabahar is an erect perennial herb and evergreen shrub. It is cultivated mainly for its alkaloids which are having anti-cancer activity. The present investigation was focused on screening of ethanol extract of Catharanthus roseus flower from environment for its antibacterial and antifungal activity against a number of pathogenic microorganisms. This study also investigated the role of environmental factors on the antimicrobial activity of Catharanthus roseus flower. Material and method: collect the plant material and its authentications. The leaves of aloe vera and flower of Catharanthus roseus were collected Badwani area and identified by botanist Dr. Vina Satya. The whole plant was cleaned and washed with distilled water after completion of the cleaning and washing activity. Plants material was collected in a beaker. Plant material were dried in laboratory room. Then plant is converted into powder form with the help of instrument and is stored in the air glass tube till future use. The first diagram is aloe vera leaves powder. Second diagram is Catharanthus roseus flower powder. Preparation of plant extract. Soxhlet extract method was used in the ethanol extract preparation of both plants. The extract was filtered with Whatman paper. The liquid was collected and stored in a glass bottle. Antimicrobial activity of aloe vera leaves and Catharanthus roseus flower. Test of specific spect microorganism of both plants. The presence antimicrobial activity test of ethanol extract of both plants was performed against one specific spect microorganism, Staphylococcus aureus ATCC number 6538. Media preparation of both plants. Always use a clean and dry flask for the preparation of media. Measure the requirement water or equivalent to purified water. Weight accurately the 30 gram of media. Slowly add media to the flask with purified water. Take care that media does not spoil. Take pH of media before sterilization and after complete of sterilization. Media was unloaded from the autoclave, cooled the 45 degree Celsius and pour media in under LAF. After sonifications of the media plate, after pH was done and growth promotion testing was performed, the media is ready for testing. Culture preparations of Staphylococcus aureus ATCC number 6538 for both plants. Handle microbial culture carefully to avoid contaminations of the area. Clean the biosafety cabinet. Under biosafety cabinet culture, Empowl open proceed further as per in-house. Incubate SCDM tubes at 30 to 35 degree Celsius for 24 hours and SDM tubes at 20 to 25 degree Celsius for 72 hours after complete. Then culture of Staphylococcus aureus ATCC number 6538 of both plants. Zone of inhibition test method for both plants. Take media plate of SCDA agar and transfer to biosafety cabinet. Take culture tube of Staphylococcus aureus ATCC number 6538 and separate plate method proceed. In each petri dish, pour 100 millions of liter of each of the ethanol extract sample solutions. Keep the place as such for 1 hour for the diffusion of solution. Transfer the plates carefully into an incubator set at 30 to 35 degrees Celsius. Incubate the petri dish at 25 degrees Celsius. Measure the diameter of the width zone produced by ethanol extract sample solutions after incubations on a suitable antibiotic zone reader or vernier calipers. Observations result, zone inhibition method result of both plants. The area diameter of zone inhibition of sample extract on gram positive bacteria Staphylococcus aureus at an ethanolic concentration by a zone of inhibition method was analyzed to assess its effect of antimicrobial. The sample extract was active against gram positive bacteria. Its observed data mean and diameter of zone inhibition of sample extract 6 cm at an ethanolic concentration on gram positive bacteria. Ethanol extract of both plants show inhibition against gram positive bacteria. The antimicrobial activity shown in both plants is due to the presence of various alkaloids, glycosides, carbohydrates, steroids, saponins, and terpenoids. It was indicated by testing. Table number 1. This is table antimicrobial activity of ethanol extract of aloe vera and Catharanthus roseus. Staphylococcus aureus ATCC number 6538, zone of inhibition aloe vera 6 cm and zone of inhibition Catharanthus roseus 6 cm. These images are microbiology result representations of aloe vera and Catharanthus roseus. Discussion. In the present study, the antimicrobial activity of aloe vera extract was evaluated and found that the hot methanol extract was very active against different types of bacteria. The extract sample of both plants demonstrated varying amounts of inhibition against Staphylococcus aureus and Pseudomonas aeruginosa. The antibacterial activity of Catharanthus roseus and aloe vera plants extract might be due to the presence of phytochemicals. These observations may be attributed to the nature of biologically active components, phytochemical, which could be more enhanced in the presence of ethanol. Conclusion. The bacterial resistance activity of the sample of extract so that Catharanthus roseus and aloe vera traditional medicine use should be maintained and scientific assessment of its active compounds should be given consider. The active compound response for the antimicrobial inhibitory activity should be identified and characterized. This extract antimicrobial inhibitory activity suggests that they could be used to treat microbial include disease. In incident time, plants and plants part have been commonly used as remedial representative for a variety of disease. The plant and plant parts are used in many herbal preparations that are found nowadays. The plants extract and plants part extract so much antimicrobial enhancement activity they can be used in many diseases. References. Thank you so much, sir.